Peter, for the introduction. My name is Jing Zhang. I'm from Royal Military College. I work as a postdoc fellow. And this talk is about insights to use the microRNAs from mammalian hair follicles to be used as a traumatic brain injury biomarkers. So traumatic brain injury, TBI, is described as any brain injury caused by physical impact to the cranium. So it is a very common disorder. Actually, it happens every day, and you can see that happening in competitive sports like a hockey game. The concussion leads to the injury. And of course, I am from a military institute, and the focus is somewhat focused on the army type of stuff, and they have the combat duties would lead to the use of explosives as weapons. So that the explosion would cause a shockwave to the head of soldiers. So that is a cause of a TBI in those type of situations. So it is very important to have some sort of system to serve as a study system as well as a diagnostic system. So, of course, the brain is very naturally used as a system to study this disorder because this is the site of the damage, but it is very hard to get access to. So people start to use blood as a biomarker system because it's relatively easier than brain to get. Still, it has to you have to have like the professional skills to take the blood. So our attention turns to the scalp hair follicle because for one reason is relatively easy to get because you just pluck the hair. Even I can do it. And also because the hair follicle actually goes through the development of the hair follicle goes through multiple cycles and multiple steps. It has it has the potential of housing rich information. So we decided to look into the potential of it to be a potential system to study TBI or serve as a biomarker system. In fact, we published our pilot study last year. So in this study, we used a microarray system platform to screen the gene expression profiles from the rat model whisker follicle to see if there's any shared gene expression patterns with brain or blood. And in fact, we found some shared results, and also because hair follicle is going through a unique process of it has its own biochemical processes, so it has also its own unique molecular signatures. So for next, there are two objectives for this talk actually. One is next step naturally is to see how sensitive the hair follicle is to different intensity of the blast. And another one is to see if there's any potential new biomarkers other than messenger RNA. We tend our attention to microRNA. So basically what we did at Suffield base is that we developed an advanced blast stimulator. So basically what this thing does is it generates a single shockwave intensity to the animal model. Basically, we stabilize the rat into the shocking chamber of this tube. We shock them with different intensity shock waves. So we tested 15, 20, 25, 30 psi intensities, and after the exposure, we collect the whisker samples from the rats. So in the lab we do microarray and of course the other molecular tests. So for this purpose, we actually follow through this flow chart. We started using a Agilent microarray platform for these follicles collected from those different intensity exposures. And then we used a subnetwork enrichment analysis to construct the messenger RNA and mRNA interaction network. So based on the messenger RNA transcript levels from the microarray data, this test proposed the potential microRNA targets that could be responsible for those changes. And then we developed a qPCR-based approach to actually amplify those potential microRNAs to see if there's any real changes in the hair follicles. Of course, we have additional tests, but that's not this talk. So from the microarray results, actually, you can see from these volcano plots, the green dots in the figures, they are the differentially expressed genes, actually, with a fold change greater than 1.5. You can see, obviously, as the increase of the blast intensity, the distribution of the quantity of the DE genes are different, obviously. And also the shared, the overlapping, the Venn diagram at the bottom shows that actually the overlapping DEGs are very scarce. So basically it says, what this thing tells us is that on the gene level, the hair follicle is able to represent, is able to respond specifically to individual intensities of the shock waves. Then we tended to a gene set level. We enriched the biological processes terms of the gene ontology terms, just to see the distribution of the enrichment. We use a page test for this test. As you can see from the heat map, the downregulation and upregulation of the GO term, obviously you can see it's different in each intensity shock waves. So at this level, we can also say that the hair follicle is able to enrich different biological processes, potentially, according to the blast intensities. So next is to see if there's any potential of microRNAs. Actually, the microRNA has been used as biomarkers because it has its own specific tissue-specific or developmental stage-specific expression profiles. And also, when it's circulating, it is usually within exosomes, so it is relatively stable to measure. And of course, they are very conserved. You can see this MIR26A, the sequence is the same across different species. So it is very easy to transfer the knowledge from the animal model to human subjects. In fact, as you can see here, of course, from this from the course of this conference, we saw a lot of studies using, exploring microRNA as biomarkers. So our strategy is to, as I said before, is to base on the microarray data on transcript on messenger RNA on the gene transcript to propose potential microRNAs, then to measure them to basically validate. So this one example is MIR183. This was proposed across all intensity shock waves. And you can see the central node is that microRNA, and you can see the blue colored shapes are the downregulated transcript levels, and red is the increase. So basically, the subnetwork says this microRNA could be responsible for these changes across all the intensity exposures. And then we measured this level, this microRNA in the hair follicle. You can see it's beautifully decreasing in all of them, and with a huge decrease in 20 psi. This microRNA has been found, according to other publications, has been found responsive to neurological disorders. So this one example is a shared microRNA candidate under the stress. And then this next one is only proposed in 15 psi exposure. And as you can see, the qPCR amplification suggests it was a decrease. And this microRNA has been proposed as a microRNA that's responsive to ER stress under central nervous system. The next example is at under 20 psi, MIR34A. This microRNA is very interesting because, first of all, it is specific to this intensity, and secondly, of course, it was downregulated in the hair follicle. And then, according to the literature, it was found once that in rat brain, it was responsive to TBI disorders. So basically what this thing tells us is that this microRNA, we can use hair follicle to detect the microRNA that's responsive to TBI. Sorry. So basically, we can use hair to get a similar microRNA pattern as a brain tissue. So basically, to summarize, what we found is that the hair follicle is, first of all, it's very easy to get. Secondly, it's sensitive to specific blast intensities. And then we explored the microRNA as a potential biomarker system. Because it's highly conservative, we can transfer the knowledge from the rat to human subject. And then we have shared microRNA. The example is MIR183, and to use the shared results, we can actually monitor the universal responses of TBI. And then we have intensity-specific microRNAs. And to use this, we can actually see or monitor the damage progression of the TBI. So basically, yeah, that is our findings. And then these are the lab members. Thanks very much.